and also a real gentleman, because I was doing something or other, and I had to run in his lab, in addition to Wetmore's lab. And so I had something that involved having to use a water bath. So I turned the water bath on because it was empty, and it had an overflow on it. So I turned it on very slowly, and went off to have dinner. No matter what Devaki said, I did not have too much to drink, but it did take a while to get back. And when I came back, I found that Thielen was paddling around in about an inch of water in his office. Well, he said, it won't be the first time it happened. He was really very kind. His graduate students were furious, and I wasn't very welcome. Except that about 4 days later, another graduate student committed what actually totally overshadowed what I had done, is he was interested in ethylene, and he had a column that was a meter long, filled with lampblack, because he was going to scour stuff out to be doing some ethylene assays. And he had the brilliant idea to test it to see that everything was tight by hooking it up with a compressed air line. Well, he did, and the top blew off it. And this mushroom cloud of lampblack hit the ceiling, and then went up this way and spread all over the entire lab. And needless to say, my little transgression was completely forgotten. I wasn't very great. Peter Rhee was in that lab at the time, and Peter said he can still open books because of lampblack. Okay, well, we got, Taylor and I managed to get part of this in a number of papers, including these three, and we published them in journal. How many people here have heard of this journal? I thought so. One. But to my knowledge, these have never been cited either. Actually, that's a lie, because this one cited that one. This one cited both of those. So to Stanford. Now, this was amazing. I was on the job market in the fall of 1954. That's a long time ago. And anyhow, the way that this was done was something that would horrify anybody today, and in fact probably bring action, legal action. Because the chairman of the department at Stanford, Victor Twitty, was an old friend of Ralph Wetmore. And they both had been president consecutively of the Growth Society, and so they knew each other. And somebody at Stanford was leaving, and so they needed to have somebody who could teach plant physiology and, of course, the mosses and ferns. So Twitty asked Wetmore whether he had anyone who was finishing up. And Wetmore said, well, yes, Winslow Briggs is finishing up. The next thing I knew, I had a telephone call from Twitty saying, could I come out and interview? So that was set up, and it was going to be a month hence. I think it was to be after Christmas. And I was sleeping late one morning, and the phone rang, and Wetmore said, there's a man named Lawrence Blinks. Blinks is a brilliant algal physiologist out at the Hopkins Marine Station. And he's here because Twitty would like to have him interview you. He's also a member of that department. So I quickly pulled on my clothes and bicycled over to the lab to find Blinks, who I'd never met before. And Blinks started out by saying, now, just what will you need by way of lab facilities? And there were a few questions like this. And finally I said, well, Dr. Blinks, I haven't been offered the position. Oh? Oh? Well, what are you supposed to teach? And I said, well, I'm supposed to give a course of mosses and ferns because Professor Steer is leaving, and I'm supposed to teach plant physiology. And Blinks said, I've been teaching plant physiology for 20 years. Oops. So anyhow, I went out, gave a seminar, and talked about ferns and leaf uncoiling and that sort of thing. And then the next day there was a sort of a tour around to meet all of the faculty members and chat with them. Now, one thing I should mention is that it's not always advantageous to stay at the same institution for three degrees. I don't regret having been at Carnegie, but there was a minor problem, and that is that they had lost a geneticist as I started graduate school, and I never had a genetics course. And then I was well into my research when they finally got a geneticist, and I was damned if I was going to take any more courses. So I never had a formal course in genetics. Well, in making the rounds of the faculty, I was introduced to Ed Tatum. And Tatum was very cordial and friendly and started to talk about what he was doing. Well, he just won the Nobel Prize. I didn't have a clue what he was up to. I had never heard of him before. I was busily wandering around amongst the osmundifrons. And, anyhow, that didn't do me any damage. So then anyhow, I wandered around, and apparently there was a rotating faculty member. So Twitty was going and saying, what do you think? What do you think? What do you think? So I had an exit interview, and I thought, well, I'll hear in a month or so, after he's interviewed some more candidates. Then he said, would you like the job? And the answer was clearly yes. Can you imagine that? There was no search, there was it was just the old boy network at its worst. Well, okay, so enough of that. That interview with Blinks, apparently Blinks never got back to Twitty, because either that or what he told Twitty was okay. Now, I started teaching, and I was teaching plant physiology, and something happened that really changed the direction of my research. And I had done my last experiments on ferns. So, this is, Fritz went back in 1926, had done a very interesting experiment having to do with phototropism. And his experiment was to shine blue light from one side, or white light in that case, and show that there was now an oxygen differential. What you did is you collected the oxygen that came out of the polar transport system into an outer block, and then there was a bioassay where you stuck the block on the shoulder of a cut-off polyptile and induced it to bend, and the bending was proportional to how much oxygen was in the block. Well, so this was the basis of the famous Kolodny-Wendt theory about lateral transport of oxygen. But you see here that we're not getting it all back. And so maybe there is some oxygen destruction going on, some photooxidation mediated by something or other. So that it was quite controversial for a while, and in fact people were able to show that light shining on an extract of pea seedlings induced the breakdown of oxygen. So anyhow, I was talking in plant physiology, and a graduate student of Tatum's, as it turns out, Jim Wilson, put his hand up and he said, you know, why hasn't anybody ever done this experiment? You know, dark, light, take a look. Does it change or doesn't it change? And then, this is the Wendt experiment. And the experiment that Jim suggested adding is this one. He said if you put a barricade in there, and it's oxygen destruction, you'll still have the differential. Whereas if it's lateral transport, you won't. So I had a senior who wanted to do a senior honors project, and he immediately took this up as a project, and by gosh, the experiment worked. You see that you just totally eliminate the differential. Now there's some loss, presumably from injury to the tissue, but it's not very large. And it's unambiguous that there isn't a differential there, whereas there is a big differential here. So that got me started on phototropism. One thing about this is that, oh, we got a paper in Science out of it. Oh yeah, I forgot, that's important. Now, one of the things about that paper that's always amused me is that we measured light intensity in meter candles. Now, I don't have a meter candle to show you, but I do have a foot candle, if anybody. We've gotten more sophisticated since then, I hope. Okay, anyhow, Bert Zimmerman came and we did detailed studies of phototropic responses of oat coleoptiles. And you can see a red light has this dramatic change. And these are different light intensities. Notice that the response is the same here at the different light intensities, but it's quite different over here at the different light intensities. And I won't go into any of the details, but we analyzed this, and then Burke worked out a kinetic scheme, which I also won't bore you with. And here, with his kinetic scheme, now this is the dots are the experimental data, and the lines are what the kinetic scheme model would give you. Actually, I showed this with great delight and pride to another giant in plant biology, and that's Sterling Hendricks of phytochrome fame,